Good morning all. I'm Dr. Subrata Das and here I am going to discuss about aquaporins. So as we know, all living organisms require water for their survival. So plants are also an important organism on earth, and here plants take the water from the soil through the root and it is transported from roots to the shoot part through xylem. So here comes the importance of aquaporin. So aquaporins are nothing but some integral transmembrane protein which makes the water channels and forms the efficient transformation of waters from cell to cell. So aquaporins are nothing but transmembrane integral protein which forms or facilitate water transportation by forming water channels. The presence of these water channels increases the permeability of cell membrane for transportation. Aquaporins are highly selective and efficient water channels which transport approximately 10 to the power 9 per second of water from cell to cell. Some aquaporins are there which are known as aquaglyceroporins, which transport water, glycerol and some other molecules as carbon dioxide, ammonia, urea across the membrane. The first water channel activity of a plant aquaporin was studied by and established by Peter Agrietel in the year 1993, and they were awarded the Nobel Prize for their discovery in the year 2003. So in plants, aquaporins are grouped into family of major intrinsic proteins, and these groups are again divided into 5 subgroups like plasma membrane intrinsic proteins, in short PIP, and tonoplast intrinsic proteins, short TIP, nodulin 26 like intrinsic protein, in short NIP, and small basic intrinsic proteins, in short SIP, and X intrinsic proteins, that is XIP. These subfamilies are again subdivided into small subgroups and consist of different isoforms like PIP, that is protoplasma membrane intrinsic proteins, consist of 2 polygenic subgroups and it consists of 13 isoforms. And in case of TIP, it consists of 5 subgroups and 10 isoforms. And in case of NIP, it consists of 7 subgroups and 9 isoforms. And in case of SIP, it consists of 2 subgroups and 3 isoforms. And in case of XIP, it consists of 3 subgroups and 2 isoforms. Structurally, the aquaporins are approximately 21 to 34 kilodaltons in size and in weight, and proteins comprising 6 transmembrane spanning alpha helices domains tilted along the plane of the membrane and linked by 5 loops, that is A, B, C, D and E. Located on the intraside, that is B and D loop are on the intraside of the membrane, and A, C and E loop are on the extracytoplasmic side of the membrane. The N and C terminal of the proteins are both exposed to the cytosolic side of the membrane. A central aqueous pore is delineated by the transmembrane